Good morning, everybody. Looks like we had some people that must have stayed up late last night. We don't have as many people in class this morning. That's okay. We'll have it recorded for them so they can keep up. I do know that a few students who are physically located still on the coast are struggling with still some power issues and just general stuff. So, our thoughts are out to them as they get through all this and try to come back to normal a little bit. 2020, man, I keep saying it. It's just been one of those years, and it just keeps on doing. Looks like we got another hurricane out there as well, but I don't know that it's going to track our way. I hope it doesn't. I hope it stays away. But should it come this way, and should things change, as you know, the university has that annoying pop-up that tells you all that's going on. So please keep that in mind as we move forward. We don't have a whole lot of classes left. We've got 2 more after today for this class because I have elected for you all not to have a final exam in this class. I want you to use that time that you would have been using to prepare for a final exam here for your other classes. So just keep that in mind that our last class will be November the 18th. And then I will be busy working to get everything graded. You will be able to know what your final grade is in Canvas as a percentage, but you won't be able to know what it officially is in SOAR until that has been released. Please don't email me asking. You can always go in Canvas and look, but I'm not supposed to give out your final grade until you all have had a chance to look at and complete your course evaluations. One thing about course evaluations that I want to mention now is some of you will be getting notices that your course evaluations are available here shortly if you haven't already. Take those very, very seriously. I can assure you that the vast majority of faculty do look at those. They do take them to heart. They are interested in your candid feedback that is constructive. Okay, so in every one of your classes, please take the time to complete that course evaluation. I use the course evaluation every year to figure out, is there some areas that I should have covered differently? Is there some areas that students felt like I didn't do a very good job on? Is there something that I could have improved on? We're always looking for ways in which we can improve, and that is your opportunity to let faculty know what you thought about things. There's a few things about etiquette. They are anonymous, so yes, you can say anything you want. But if you want your opinion to be taken seriously by a faculty member or by that faculty member's school director, you need to be factual. You need to be polite. You need to be respectful. And you can say exactly what you want. If things went really, really well, let them know that you appreciated that. Give them some good positive feedback. If you think things could have been better, tell them the areas that you think that they could have done a better job with. They will take it to heart. I promise you. The vast majority of faculty, at least, will that I'm aware of. So it is your opportunity to have your opinions heard by the faculty in a way that is anonymous. So it is anonymous. Faculty cannot see their course evaluations until after grades have been posted. So you don't need to worry about any type of ability for a faculty member to trace it back to an individual. Okay. Having said that, I will have students quite frequently that will tip their hand to who they are in their comments. You're free to do that, but just note that won't be anonymous. Right? So if you type your name in the comments, that does not get edited out. Okay, so the comments go to the faculty member in raw format. So whatever you type in there, that's fine. So if you want your faculty member to know, type your name. You can do that, but I highly recommend that you keep it anonymous and honest and polite and respectful. Okay, so for today, we've got a few things that we're going to cover. What happened with my screen? First thing is research opportunities with Dr. Rangachari. How would you highlight or how would you summarize Dr. Rangachari's research area? What kind of research does he do?
Biochemistry with like proteins and collagen.
That's right. So he is a biochemist by practice. Dr. Rangachari has an interesting career path to biochemistry. His master's degree, if I remember correctly, is actually in organic chemistry. And then on for his PhD in more bio-relevant kind of chemistry, biochemistry, if you will. And then moved into, with his postdoc at the Mayo Clinic in Florida, was more biophysical. Okay, so he is a biochemist. It's a big umbrella kind of thing, but really he's a biophysical chemist, meaning that he studies physical attributes of biomolecules. Okay, so what's his main biomolecule that he's interested in? Or his main disease thing that he's looking at?
Proteins.
Okay, so he's looking at proteins, but proteins related to what kind of disease state? Neurological, yeah. So he has this neurochemistry kind of bend, and he looks an awful lot at things like what are called A beta, which is responsible for, anybody know?
Prion disease?
Okay, so yeah, prions, yeah, and so it is largely Alzheimer's, Parkinson's type diseases, things that rely on the self-replicating entities to form these toxic plaques that lead to these neurodegenerative diseases. Many of you in this room probably know somebody who has suffered from Parkinson's and or Alzheimer's disease. I know I certainly have acquaintances, family friends, family members who, as they have been aging, have had to deal with these issues. 10, 15 years ago, things were a lot more dire for those individuals than they are today. We now have treatments that can help prolong their lives. And in a much better state. I can tell you, my father-in-law suffers from Parkinson's type disease. He takes these injections that now allow him to function relatively well. Certainly not the same as he would have been functioning 20 years ago, but he's not in a debilitated state anymore. So studying these types of diseases, how these proteins interact, how these proteins form these plaques, that's a biophysical process. What interactions are going on between the proteins so that they form these fibrils? How do these fibrils interact so that they form these plaques? How do these plaques interact with other tissues? All of this stuff is important basic information or basic science that we need to know before we can even think about making good drugs. We've got to know how these things work. And so Dr. Rangachari spent an awful lot of time talking in the video about his general research area, right? If you were to go and do research in his laboratory, you're going to be in a truly multidisciplinary area. You're going to be dealing with biomolecules, proteins. You're going to have to figure out how to purify those proteins, how to isolate those proteins, how to study those proteins. That requires a variety of techniques. You can't simply just get a sample of a protein and look at it with your eyes. You have to interrogate that protein with a variety of different techniques, different spectroscopies, if you will. Does anybody know what a spectroscopy is? What is spectroscopy? Damien? Yeah, that's true. I mean, so we can use spectroscopy to determine those types of things, absolutely. But spectroscopy in itself is a technique that typically we're looking at how the matter interacts with some form of energy. Okay, and when we do that, we can determine how it's made up. We can determine what interactions are there. And so Dr. Rangachari does an awful lot of that. So they'll take these samples, they'll shine light on them, so to speak, and they'll see how that light interacts, and that can tell you whether or not they formed these fibrils, these plaques, whether they formed helices, all different kinds of different shapes, and they can start to study what's going on. So they might start in a cell, grow up some of these proteins, study them in the cell or study them outside of the cell, and get some good information on what's going on. I can tell you that Dr. Rangachari collaborates with a variety of people on campus. He collaborates with other biochemists. He collaborates with biologists. He's collaborated with me a time or two. We've made some small molecules in my organic lab that he could use to see whether or not those small molecules would inhibit the formation of these fibrils or these plaques by interrupting some of those interactions. He works with folks over in the polymer program. Why would somebody in biochemistry be working with somebody that deals with polymers? Matt?
Because proteins are a type of polymer.
That's right. Proteins are nothing more than an organic molecule that are linked together through what are called amide linkages. Y'all don't need to know this. I'm not going to quiz you on it. Right? And they form biopolymers. Right? And so working with folks that have techniques that can deal with these so-called macromolecules is helpful for him. Right? And so these collaborations are very, very fruitful. And so I can tell you from firsthand knowledge, because I have worked with Dr. Rangachari off and on over the years, that his undergraduate students do come away with an awful lot of skills that they can then apply if they decide to go to medical school, if they decide to just go to graduate school, if they decide to just get a job. So every skill that you learn in all of these labs, really, you're going to come away with a good set of skills. But I know many of you are kind of bio interested, if you will, right? And so, Dr. Rangachari is an example of a biochemist at the institution, and that I think really speaks to a lot of kids, because he's dealing with a disease state that many of you at least have some level of knowledge about. You may have a family member that's dealing with these things, and that sparks your interest in studying these things and knowing more about them. Anything that caught your eye of what Dr. Rangachari had to share with you? Damien?
I don't know, some like how recent some of these like findings were, like in 2010 or 25, like how recent everything was, and I think that like everything just could happen just like then, and would be where we are now, stuff like that.
Yeah, that's right. So in the biotechnology, biopolymer, whatever you want to call it, fields, a lot of times things will kind of go along slowly, and then there's a discovery, and everything just opens up, and things seem to accelerate very, very quickly for a period of time. We're there with a lot of these prion-type diseases. There have been recent discoveries, and by recent I mean 4 or 5 years, or even maybe a little longer. For you, that may not seem very recent, but in a scientific sense, it is a very recent set of discoveries that have really changed the way people started to think about how these things work and interact, and how you might be able to inhibit the process in a way that is druggable. And that's very important to understand. Chemists and biochemists will go into the lab and probe something, and you might have something that looks like it will kill cancer cells or keep A beta from forming fibrils and toxic plaques, these so-called amyloids. But the molecule itself isn't druggable. That is to say, we can't give it to you, take it as a pill, and it get to where it needs to be to do the work. And so we're always faced with that of, we found something, we've learned something new, but this isn't going to function as a drug. It can't cross the blood-brain barrier, for example. So then you go back to the drawing board, and you know something, and now you think about how can you make that molecule druggable. And so there's always this back and forth to try to get to medicines and different things like that takes a long time. It takes years to get a drug to market, so to speak, or to even find a drug. All right, so this was our interview with a former Golden Eagle, Ariel Kelly. Where's Ariel at right now? She's at the University of Alabama. And she is going to be defending her PhD soon. Does anybody know how soon?
December 1st, isn't it?
That's right. Just in a little less than a month, she'll be defending her PhD. Ariel Kelly graduated from here, I think, 4 or 5 years ago now, and went to Alabama. She went to Alabama at the same time that Christina Probasco went to the University of Alabama. Christina, of course, recently graduated, as you all will remember, and now has a job with Thermo Fisher. And Ariel is looking to graduate. What's she want for her career path? Anybody remember?
She wants to be a professor, right? She wants to do the academic side of it.
Yeah, she wants to be on the academic side of things, right? And so she's thinking about teaching as a profession. She's thinking about maybe doing some research. What kinds of levels are there for a person with a PhD that might want to go into academics? Does everybody have to go to a place like USM to be in academics? What other kinds of institutions could they go to? USM is what we call an R1 institution. We're a research-intensive institution. So yes, we teach, we do other things, but we are primarily a research institution in a lot of respects. But is every institution that way? No. There are institutions that are R2s or R3s that maybe have some research, but don't focus on it as kind of the main theme of as an institution. There are 4-year institutions that offer no graduate degrees. We offer graduate degrees here, obviously, masters and PhDs, in everything from the humanities all the way into the sciences, right? So research is definitely a central part, and graduate education is definitely a central part of what we do, in addition to the undergraduate research or teaching mission that we have. But there are institutions across the state, across the country, right, that focus primarily on undergraduate students. There are private institutions that only focus on undergraduate students. And so if you're really interested in more of the teaching side of things, you would end up going to one of those institutions. Having said that, almost all institutions, with the exception of community colleges, require some level of research if you're in the academic field. Okay? So here, when I went up for my tenure after 5 years of being an assistant professor, primarily they looked at my research accomplishments. My teaching accomplishments were good to have, and I needed to do well at it, but primarily they looked at my research accomplishments. Did I get any grants funded? Did I publish any papers? Did I train graduate students? Did I get them graduated? They looked at those types of things. At an undergraduate institution, they look primarily at your teaching successes, right? So how many classes have you taught? What were your teaching evaluations? Again, an important thing to remember, right? We faculty are judged to some part on your teaching evaluations. And then secondarily, they'll look at, did this person do some research? Did they get some papers published? But primarily they're looking at the research at those types of institutions from a training of undergraduates perspective. How many undergraduate students did Dr. Masterson work with, and did they graduate, and did they write a thesis or a senior project? Those types of things. So it's a little different mission. And then you have the community college, which is, I would say, the vast majority of what they look at is teaching, maybe a little bit of service, and zero research for the most part. And so there are 3 different types or flavors, if you will, of institutions across the country that she might be able to choose from. Anybody get a hint as to which one she was kind of aiming for? Did she talk about wanting to do a lot of research, or did she talk about being more interested in teaching? Damien?
I want to do more teaching and less research.
That's right. So she wanted to do more on the teaching side. And so you will remember from last week that Shelby Dickerson got an opportunity to teach, and from that opportunity decided she might want to go into industry. So it's very, very important early on to determine whether or not you think you're going to like the academic lifestyle. Some of you will probably choose to go the academic route. Some of you will shy away from it. Some of you will want to go to medical school. Some of you will end up going to medical school and decide that you want to do medical research and maybe teach medicine. There are a number of possible avenues for you, again, with a chemistry or biochemistry degree. And so I hope you've seen through the progression of the folks that I have shown you that were former Golden Eagles, many people have gone many different paths. That was the goal of what I wanted to show you all, was that with a good solid education in the chemical sciences, there are many paths for you. And so as you mature as a college student, you can still choose which path you can go down fairly easily. Okay? So not everybody has to become a chemistry teacher. Not everybody has to work in industry. Not everybody has to work at a government lab, whatever the case may be. Any other final thoughts about Ariel Kelly? Okay. All right, this brings us to Le Chatelier's Principle from the chemistry book. Henry Louis Le Chatelier, and I'm probably pronouncing Le Chatelier wrong, but that's how I pronounce it. This has been a principle that's been around for a long time. It is one of the things that for you as a chemist and or biochemist, you need to have a strong understanding of Le Chatelier's principle. Le Chatelier's principle can help you explain so many things. So many things. Okay? What is Le Chatelier's principle if you had to go tell your grandma what it was? How would you distill it down to a note card? What is Le Chatelier's principle?
If you have a reaction that goes to equilibrium, and you have it at equilibrium, if you change any of the factors, temperature, concentration of products or reactants or whatever, the equilibrium will change to offset that change in conditions.
That's right. Equilibrium. What do we say when we call it KEQ? What is that?
The equilibrium constant.
That is the equilibrium constant, right? And I promised y'all I wasn't going to teach a lot of content, and I'm really not. I'm not going to test you on any of this. But I think Le Chatelier's principle is so important, okay? Especially for those of you when you get to organic chemistry. You're going to be able to understand how we can take a reaction and get a complete conversion to product if you understand Le Chatelier's principle. So let me give you an example. Let's suppose you take something like vinegar, which is acetic acid, which is what I've just drawn here. Don't worry about the structure if it's not clear to you. Just take it as A if you want to, okay? You can take things like vinegar, and you can react them with things like ethyl alcohol, and they will react in equilibrium, and it will form what's called an ester. In a molecule of water. Right? And so in theory, we could say, we could call this A, we can call this B, we can call this C, and we can call this D. Right? And we can then express that as an equilibrium reaction. It is an equilibrium. I can take that A, I can put in B, I can let it sit long enough, and I can start to see C and D form in what's known as a condensation reaction. Okay? And this has an equilibrium constant. And we know that the equilibrium constant is always products over reactants, right? Or you will know that when you get to that part in general chemistry. So it's the concentration of the products divided by the concentration of the reactants. What does it mean to be at equilibrium?
The rates are equal in both directions.
That's right. So as A and B form C and D is decomposing to form A and B at exactly the same rate. And so what does that mean? That means if Mac was to run that reaction and he came in, if it was at equilibrium and he looked at it today, he would notice that A and B and C and D are some concentration. And he would come back at some other time in the future and look at it again. He would recognize that none of that has changed. That's what it means to be at equilibrium. The concentration of the stuff isn't changing with time. It is changing, but it's changing at the same rate such that you can't tell the difference. Okay? Let's say you wanted to convert all of A into C. You're working at a company, and they take vinegar and they make this ester out of it. Esters smell good. They make perfumes out of them. You're working at a perfume company maybe. And you want to take all of this and convert it into that. Can you do that in an equilibrium reaction just by itself?
No.
No. Not in this particular reaction anyway, because the equilibrium doesn't completely lie to the right. So, but we can use Le Chatelier's principle to our advantage. So what could I do?
Remove water.
I could remove water, right? Water's pretty easy to remove from a reaction. We can distill it out, for example. So if I remove water from the reaction, what happens to the equilibrium? Does it shift to the right or to the left?
To the right.
It shifts to the right. And so as I continue to remove water from the reaction, I will convert eventually all of A into C. And my boss will be very happy and he will pay me my paycheck and we will be good to go. Okay? Is that the only thing I can do?
Depending on whether it's exothermic or endothermic, you could change the temperature.
That's right, I could change the temperature. I could change the temperature and drive the reaction one way or the other. I have to know whether or not the reaction is exothermic or endothermic. This reaction, just by looking at it, it's hard to know because the bonds that are changing are very similar. Okay, you'll learn more about that and don't panic. You'll learn more about the thermodynamics when you get to Chemistry 107. But is there something else I could do? I could do what you suggest, but I don't really know right off the top of my head what the thermodynamics are, so let's think about something else I could do.
You could add more of the reactants.
I could. I could add a lot more B. So maybe we've got a source of this alcohol that's really, really cheap. So I could add more of this, and so as I increase the concentration of this, that's also going to push the equilibrium that way, right? Le Chatelier's principle is about a teeter-totter, or a balance. You've got a fulcrum, you've got a board, you've got two weights, and it's equal. When I do something on one side that makes it unequal, it's going to try to regain that balance. Hence equilibrium, right? It's going to try to regain that KEQ value. Okay? I don't have to do any of the math to know how I can push the equilibrium to the right or to the left. In 107, you will learn how to do the math, and that's going to be helpful for you. But when you're in the lab working, most of the time you're not sitting there with a calculator doing the math. You're using what we call chemical intuition to try to determine how to make the reaction work the way that you want. Okay? You all don't think about the words that you use. They come off of your head very quickly as you have thoughts because they're ingrained. Eventually you will have chemical intuition ingrained in you to where you will know by looking at a reaction which way the equilibrium lies, and you'll know how to disrupt that to do what you want. Maybe Damien has a bunch of C and he wants to make A. Well, it's the same reaction, he just wants to do it in reverse, right? From what we talked about. So how would he reverse that reaction? Add water. That's cheap, right? Just turn on the faucet. Add a bunch of water to it and watch the equilibrium go the other way. He could then isolate A, make his boss happy, get his paycheck, and go on his merry way, right? You can go both ways with these processes. Le Chatelier's principle is also important in biochemistry. Most reactions that make living things live are equilibrium reactions. And what's happening, the reason that we move in the directions that we move and function the way that we function is because those products that are formed are consumed by some other reaction, which shifts the reaction to the right. Okay? Very important in neurochemistry, for example. Neurotransmitters interact with receptors in your brain, right? And that's an equilibrium process, and you can shift it one way or the other. And so for those individuals who have issues with those receptors and those drugs or those molecules binding, we can disrupt that with or improve that by using Le Chatelier's principle by giving them things like serotonin and other neuro drugs that will shift that equilibrium and help more of it bind. We can do that kind of work. Okay? So again, Le Chatelier's principle is a very, very important concept. And here's an example with the nitrogen oxides using temperature. So high temperature, room temperature, and low temperature. You can clearly see the differences in colors because the equilibrium is being shifted between one form or the other. Okay. So what did I tell you last time was a very, very important concept that you needed to remember?
Acids and bases.
Acids and bases are equilibrium processes, right? You have Kas and you have all of these types of things that you're going to be dealing with acids and bases. Le Chatelier's principle comes into play there as well. These are all related, and so I want you to understand that. Let's talk about polywater. Why do you all think I gave you this one?
To highlight the importance of the scientific method.
Exactly. This is a classic example of the scientific method at work. Tell me a little bit about what you read about polywater. This actually was a controversy that came out before I was born, so it's pretty old. What do you know about it? Were the observations flawed? Were they real?
It was a situation where like they found this water and like with crystal structure in there. They thought it was a new type of water because it had different properties than real water. And you know they said they did they said they could replicate it, but then at the same time some scientists could, some couldn't. And then you get to the like authentic studies because like it was in quotes and it says like what they said they did, they didn't really do, and like it was being proved, that it wasn't.
Yeah. The observations that these folks were seeing, there's no doubt that they witnessed these observations, that they were forming these little bubbles of what they were calling polywater at the time. It looked like it had different densities. It looked like it had different boiling points. It looked like it had different melting points. It looked like very, very different form of water. But there wasn't quite something right about it, was there? Some people would study it and replicate the result and say, we can verify that. Other people would be like, no, not at all, right? One of the central things for the scientific method is that we share our results. We allow people to interrogate those results through additional experiment. That's what you're seeing played out here, right? I mean, it's pretty sensational when you read this, right? That people were kind of worried about, if this got out into nature and all of the water became polywater, life as we know it might end. There were people that believed this stuff, educated individuals. You kind of believe this. We can look back on it now as if, oh, how silly it was. But I did a little more research on polywater. I'm certainly not a polywater expert, okay? But, the federal government got involved, started giving lots of research grants to study polywater. They were worried about a polywater gap between us and the Soviets. Heaven forbid we have a polywater gap. We were studying this stuff pretty intently. And it turns out it was nothing more than what? Contaminated water. Ultimately, when the final experiment that put the nail in the coffin for this, they couldn't distinguish polywater from human sweat. And yet, the people who did this type of work were doing what they thought was thorough. They were reporting the results. They were making some pretty sensational claims. One of the things that I want you to understand as scientists, not just chemists, but as scientists, it is extremely important for scientists to be skeptical. That does not mean be rude, but you also have to have an open mind. You have to have an open mind to the new possibilities, but you need to be skeptical enough to interrogate the results of each and every scientist. When you read a scientific paper, you should question that. You should question the facts that we give you in class. You should interrogate them with questions to your professors. The things that we teach you in class today may not be the same things that we're teaching students 30 years from now. Certainly will not be the same thing. Can you imagine what a biochemistry book looked like in the 1940s? Before we knew about DNA? Does that mean that textbook from the 40s was wrong? It was incomplete. Science can be incomplete. And we make the best decisions that we can with the information that we have at the time. That's what we do. That's the scientific method. Polywater is an example where somebody made an observation, extrapolated it to a sensational claim of, oh my gosh, if this gets out of this capillary tube and into the oceans, we just might have polywater everywhere. And when you think about that now, especially after you all get through physical chemistry, you all will look back on that and go, that made absolutely no sense logically. We can be fooled. And that's why it's so important that we have an open system of scientific inquiry and we allow people to challenge our results with additional experiments. Okay? So polywater, again, happened before my time, but it is one of the classic examples that made it into the chemistry book as a defining moment. And so I think this is a perfect example of showing the validity of the scientific method, the success of the scientific method, at least in my mind. All right, the last thing I gave you was a case for cold fusion. There were several slides that you could go through and look at this. Who were the individuals that were involved in the cold fusion controversy? You remember the last names? Pons and Fleischmann. Okay? I was around for this one. I was born. I was around. I was actually in community college when this started to become an issue. What is the allure of cold fusion? Why would something like that be important, Damien?
You can a lot of energy in an easy way, just basically.
That's right. Getting energy in a very renewable and easy way. Is that a sensational claim? It is. It almost sounds like something for nothing. Right? At room temperature, I take this metal, I apply a little bit of current, and all of a sudden I get more energy out of it than I put into it. The hairs on the back of your ear should stand up and go, I'm going to be skeptical about that. Okay? Yes, people are still studying the concepts of cold fusion. We would like to be able to have fusion. You can take two nuclei, fuse them together, get energy. This is how the sun works. The sun works on fusion. Right? We could take seawater, the deuterium in seawater, form hydrogen with it, and get energy. Okay? This was like polywater. If you look through this, though, there were some issues that came up with Pons and Fleischmann that really caused a lot of people concern. They got some funding to study this. They had an agreement with another scientist that they would do independent research and do some things to see if they could replicate the results. Pons and Fleischmann jumped ahead of that agreement and went ahead and published without it actually being vetted very well. And it made it into journals, some very good journals, that then we had to step back from and went, on further looking at this, there's no fusion going on. Pons and Fleischmann were looking at the heat that was evolved from their apparatus. But for those of you who know anything about nuclear reactions, there are these byproducts called radioactivity, right, that comes off. Neutrons should have been coming off of this reaction, and they couldn't detect any neutrons at all. Well, if it's not doing that, you can't have fusion, right? And so, again, this is another example where, unfortunately, the careers of two well-established scientists was tarnished, because they started to believe their own hype about the possibility as opposed to giving it time. I mean, they saw something that they couldn't quite explain, and it needed to be investigated. But they made a quick jump to, must be cold fusion. We've solved the energy crisis with a beaker full of heavy water, a little palladium metal, and a 9-volt battery. That just sounds too good to be true, doesn't it? Probably is. Right? And so that, again, is the take-home message here, that the scientific method, I think, serves us very well in all things. Anything that you can apply the scientific method to, I think you should, as a citizen. When somebody tells you something, you should look at it skeptically. You should want to see evidence and proof. And you should want to be able to make up your mind on your own. Scientists eventually come to things called consensus. Climate change is one of those things, right? Climate change, we have come to a consensus from the majority of the scientific data that we believe that human activity is responsible for global climate change. But we could do an experiment that can show that that's completely falsified. We have not completely proven that hypothesis, but the vast majority of the data suggests that that is the case. Okay? So should we be taking actions right now based on the best data we have, or should we just be waiting 100 or 200 years down the road? No, we should be taking some action, right? And this isn't political. But we've got enough information to know that certain things are happening, and we can test that. We can change some things, right? We can do that type of work. But you should always, as a scientist, have a skeptical eye on any claim that anybody makes. Okay? You don't have to believe anything at face value. You have a right as a scientist and as a citizen to expect that those who are doing these types of studies, whether we're doing studies on drugs and cures, you should be able to find that information and study that and determine whether or not the data makes sense. So again, I want you to leave this class with a good understanding of the scientific method, because you're not going to get a whole lot of that after this class. You're going to get a lot of scientific content, things that have been tested and tested and tested and tested. The stuff that we teach you as an undergraduate, stuff like acid-base chemistry, thermodynamics, it has been tested and tested and tested and tested, and it holds up. Holds up so much that we call them facts. 10 years from now, though, somebody may come up with a different theory that explains things a lot better than the theories we have now, and we will have to change our textbooks again. Okay? But all of you, if you remain a chemistry major, a biochemistry major, will one day have to step into a research lab to graduate. And you will be working on a project that has not been tested and tested and tested and tested. You will be working in an area that is cutting edge. And by cutting edge, I don't mean that it's going to transform society, but it's cutting edge in the sense that it's been unexplored. If you go work in Dr. Rangachari's lab, you will be doing experiments that nobody else has done before. You will be getting data that you should look at skeptically. We found the cure to Alzheimer's disease. You should be very skeptical about that. You should want to test it further. You form a hypothesis. You try to prove or disprove a hypothesis.
You attempt to disprove it.
You attempt to disprove it. The other way gets biased. Right? Oh, if I do this, that would prove my hypothesis. I want to find an experiment that could disprove my hypothesis. Right? And so when you get into your research labs and start doing that, you really need to look at all of your results with that skeptical eye. Especially if it's especially when it's a fantastic claim. And I've been in laboratory settings where fantastic claims have been made and where fantastic claims have been shot down. 99% of the time, a fantastic claim will be shown not to be the correct answer. I was in a lab one time where the laboratory researchers thought that they had probably found the root cause of atherosclerosis, the hardening of the arteries. It was a wild claim. The initial data seemed to point to it. Further experimentation showed that while it was a contributing factor, it was not the cause. I was at the University of Oklahoma when one of the scientists there thought that they had figured out how Alzheimer's amyloids propagated. And if they had figured that out, they would have been at the forefront of curing Alzheimer's disease. We're still talking about Alzheimer's disease 25 years later. Obviously, that extraordinary claim was not the case. But in all cases, those scientists are still doing good work, right? And the scientific method allowed them to correct the record. So again, it's very, very important that you have a working understanding of that. All right, well, I'm going to give you 10 minutes back today. Your attendance assignment is available now in Canvas. Go in and complete that. And then, of course, your notebook reflection due by tomorrow night at 11:59. I will see you all next week. Next week we will have a visitor, at least virtually, from the Office of Nationally Competitive Scholarships. So I'm going to dedicate maybe 20, 25 minutes for her to talk to you about being applicants to some of our nationally competitive scholarships. And I want you to think seriously about, as you move along, applying for things like the Goldwater, the National Science Foundation Awards, those types of things, because I think many of you will be competitive for those. So please be here to ask her any questions that you may have about her office and that process. Have a great day.